Okay, Hitoshi, it's 10 o'clock. Shall we start?
Okay, sure.
Okay.
Okay, hi everyone. I'm Hitoshi from Japan. I will chair this session. First, sorry, I have a trouble with my left eye, and it's a bit hard to keep it open. So sometimes I will close my eyes, but I don't get asleep. But I'm hearing your presentation and comments. So today we have a beam damage working group report. Speaker is Dr. Arun Asundi from Slack, and he will present the outcome of beam damage working group. There are nearly 20 contributors. Appreciate it. And you're encouraged to give your questions or comment in the chat. During break or after his talk, we will have a question and discussion session. So Arun, could you start your share your slides and start your presentation?
Yes.
We see your desktop now. Yeah,
Right.
that's fine.
This looks
That's
correct?
Great.
Okay, good. All right, thanks, Hitoshi. Yes, my name is Arun Asundi. I'm a researcher at the Stanford Synchrotron Radiation Light Source, and I'll be sharing the results from the working group on beam damage and X-ray spectroscopy. This report was prepared by myself, Mekin Abernathy, and Riti Sarangi at SSRL, and it includes contributions from an international group of experts in spectroscopy and synchrotron science listed here. So we approach this topic by considering 5 areas relevant to beam damage. These are definitions and mechanisms of beam damage, how researchers can identify and mitigate the effects of beam damage, how data processing procedures and interpretation of results are affected, standards for reporting results, and finally, an outlook on how beam damage will need to be addressed in the next generation of X-ray sources and experimental stations. In the past few months, we worked with the experts listed on the previous slide to get their thoughts and experiences on these topics. We hosted 2-hour Zoom discussions to go through their ideas in detail, and they're providing feedback on the final report that we're putting together. From this working group's activities, we will have this presentation summarizing our results. We're preparing a written report to be published that includes our recommendations related to beam damage. And to make the results very concrete and immediately accessible, we've distilled our results into 5 recommendations for users and beamline scientists to apply to their work. So I'll start with the definitions and mechanisms of beam damage. We define beam damage or radiation damage as any undesirable X-ray dose-dependent change in the sample or sample environment. This can take many forms. The sample can undergo chemical changes. One of the most common is photoreduction. So at the bottom left is an example of the photoreduction of a copper-2 species to copper-1 as a function of X-ray exposure time. There can be physical changes to the sample, like nanoparticle agglomeration, changes in crystallinity, or dissolution of the active species into a solvent. The beam can also induce undesirable processes like photocatalysis or metal ligand charge transfer. And finally, we also include changes to the sample environment, like deposition of material on a capillary wall or X-ray window, damage or puncturing of an X-ray window, or degradation of other species in the sample environment that compromise the integrity of the experiment. It's important to note that even very robust samples can be susceptible to beam damage at high enough flux density and high enough exposure times. So researchers should always be considering beam damage and checking for it when they do their measurements. Now, shown as on the previous slide, beam damage can take many forms, and this makes it very challenging to model beam damage processes, especially since multiple processes can occur simultaneously. One known mechanism is through the generation of oxidizing and reducing agents. For example, in aqueous samples, the beam can induce formation of solvated electrons and hydroxyl radicals, which can undergo redox reactions with the species of interest. More generally, the dose-damage relationship depends on many factors. The sample environment plays an important role. This at the top right is an example showing the effect of glycerol in the sample on the rate of photoreduction of copper-2 species. The beam configuration is also extremely important. For example, at the bottom right, this is a study showing that at high beam flux densities, the researchers saw agglomeration of rhodium nanoparticles, whereas this behavior was not observed at lower flux. The X-ray energy also relates to the absorption cross-section with higher absorption at lower energies, and thus beam damage tends to be more severe in the soft and tender X-ray regimes. And finally, the detection method can play a role. For instance, if beam damage process alters the surface of a sample more than the bulk, this will be more readily observed using a surface-sensitive detection method. So next, I'll summarize our recommendations for users. Our first recommendation is to know your sample. Users should come to the beamline with some knowledge about the structure and chemical properties of their sample, and they should have some idea from the literature or previous measurements of the expected spectral features, like intensity and energy of features in the pre-edge, rising edge, and white line. For in situ measurements, where the user usually expects to see some change in their sample, they should know what the starting material is and what changes are expected during the experiment under different conditions. For example, at the bottom right here, this shows the reduction of rhodium oxide nanoparticles in hydrogen. The blue curve is showing the evolution of water during a lab TPR experiment. You can see the reduction is happening around 80 degrees Celsius. However, when the same experiment on the same sample was performed at the synchrotron, the reduction occurred around 30 to 40 degrees. And so this is an example where the user should come to the beamline having already done the lab-based TPR measurement. So if they see some discrepancy in the behavior, they can investigate further if this is the result of some beam damage process. Next, there are several steps that users should take to identify whether a sample is experiencing beam damage, and methods to mitigate it if it is. Before the measurement, the user should consider the possibility of beam damage and allocate time in their proposal request for beam damage studies, especially if the behavior of the sample is relatively unknown. The user should discuss the sample preparation with the beamline scientists so that the data can be collected without excessive sample exposure. So for example, if the sample is too dilute, the user may need to collect more scans for longer time than would have been necessary with better sample preparation. It's also worth considering alternate methods. For instance, it can be challenging to collect oxygen K-edge spectra due to the high absorption cross-section at low energies. So the user may consider hard X-ray Raman as a potential alternate technique with lower X-ray dose. During the measurement, the user should perform checks to see if the sample is experiencing beam damage. So one important metric to watch out for is the edge step to see if it changes over time, as this may be a sign of particle agglomeration or dissolution of a species. The user should check for beam damage before starting data collection. One way to do this is by choosing a few energies of sensitive features in the Zanes region, like the pre-edge, the edge, or the white line, and measuring a time series of the absorption at those energies for the full duration of a scan. You can see an example of this here at the bottom right, showing the changes in absorption at a fixed energy on the copper K-edge, with different thicknesses of attenuating filters in front of the sample. This method is especially useful if beam damage occurs very rapidly. If the user immediately just starts collecting data, then the damage may occur in the very first data point of the pre-edge background. And by the time the scan reaches the Zanes region, the users may not have realized that the sample is already damaged. If the rate of damage is slow, then the user may compare multiple spectra collected on the same sample spot. Or another option when collecting multiple spectra on a single spot is to periodically move to a new spot and collect a quick low-resolution Zanes scan to ensure that the spot being measured is representative of the sample as a whole. It's also important to note here that the data needs to be collected with sufficient signal to noise so that changes in the spectrum can be readily observed. All right, next we move on to data processing and interpretation. One thing we emphasize is the importance of assessing beam damage before and during the measurement. The user should not discover that the sample was beam damaged during their data processing. At that point, it's probably too late. If the user has taken the appropriate steps to mitigate beam damage and it was still not possible to fully eliminate it, then the effects of on the sample must be explicitly addressed in their data processing. The user should collect a dose-dependent series of spectra and look at features whose energy and intensity change to estimate the extent of photo damage. You can see a nice example here where the researchers looked at changes in the pre-edge intensity and edge energy and compared with references to understand how the sample changed and how these features likely looked in the original sample. If possible, the user should also measure a fully damaged sample to understand the maximum changes in the spectrum and potentially use this for deconvolution to estimate what a zero exposure spectrum would look like. Lastly, the users can support their analyses with computations, especially if it's been shown that there's a good match between computation and experiment in the type of system that the researcher is studying. If a user sample is damaged by the beam, we also recommend they use complementary techniques to determine the effects on the sample and quantify the extent of change. Some of this can be done with model species, especially if the amount of sample is limited or not directly amenable to these other techniques. Research can use a variety of spectroscopies or physical characterization techniques to assess the extent of sample change. And in some cases, these methods may be built into the beamlines themselves. Otherwise, the user can measure the sample before and after X-ray exposure to understand how the sample has changed and what the extent of the beam damage was. Lastly, for users, we recommend some details that should be included when reporting the data. One challenge in the community is that beam damage is often not reported at all. So we encourage researchers to describe how they assessed beam damage and any mitigation methods they used, even if the final data sets were collected under damage-free conditions. The goal here is to provide enough details so a reader could reproduce the measurement, and if the reader is planning measurements on a similar system, then they can understand what steps they may need to take in order to obtain good quality data. We suggest including details like the flux density, exposure time, methods used to assess and mitigate beam damage, and some evidence, perhaps a figure in the supporting information showing that the data was collected without any sample damage. Additional details that would be useful to include are descriptions of figures or figures of unsuccessful measurement attempts, dose-dependent series of spectra, and details of the beamline configuration. Some journals include a laboratory notes section, which is a good place to include these details. All right, so next I'll move on to our recommendations for beamline scientists. Our first recommendation is to understand the beam characteristics. Going back to our definition of beam damage, it all starts with the X-ray dose. So it's important to measure the flux density and characterize the spot size and shape to accurately quantify the X-ray dose on the sample. The variations in flux with energy should also be characterized. Flux can be measured with photodiodes or ion chambers with advantages and disadvantages for each method. With ion chambers, the flux can be measured simultaneously with data collection. However, with very high flux beamlines, it may be necessary to fill the ion chambers with helium, resulting in low absorption and a very high sensitivity to small perturbations. Additionally, with high flux, ion chambers can be sensitive to Compton scattering, which leads to nonlinearities in the response to flux. Photodiodes have an advantage of being able to be positioned at the sample location, but they cannot be measured simultaneously with the data collection. We encourage beamline scientists to take the lead here on characterizing and documenting the beam characteristics, since many users may not have the time or the expertise to do so. This can be done during the setup, and then the beamline can be handed over to the user with details on the flux and spot size. We suggest that the beamline scientists communicate these few key metrics to the user so the user can easily understand and include that in their notes. We also suggest including metadata like ion chamber readings into the data files automatically. And then this can be compiled at a later date to assess changes in the flux density over longer time periods. Beamline scientists also have a responsibility to develop and maintain robust sample environments. Any hardware that's supplied to the user should be characterized, for instance, by reproducing published data. And as we've talked about, trace impurities like water can result in radical formation that affects the sample, and these should be considered when developing and maintaining sample environments. If it's possible and relevant, it can be useful to accommodate beam-off measurements using the same hardware so the user can compare the sample behavior with and without the beam, without having to sacrifice their precious beam time. For instance, with a catalysis measurement, if the user can analyze the reaction products with and without the beam, either by collecting a sample or if the analytics are built into the reaction system, then making this available to the user outside of their measurement time will allow them to ensure that the beam on and beam off behaviors are the same. We also recommend that some options for beamline optimization, like methods to adjust the flux, are made available to the user, and the beamline scientist should educate the user on how and when to use these options. Alternatively, the beamline scientist may request a representative sample from the user and assess beam damage during setup. They can then hand over the beamline to the user with a clear recommendation on how to collect damage-free data. When developing beamline hardware and software, it's important to also consider the dark period. Some beamlines include shutters to limit the exposure of the sample when data is not being collected, but the diffusion of radical species, which can lead to these oxidation and reduction processes, continues even when the beam is off. So fast hardware and software are needed to reduce these dead times. Lastly, we emphasize that the beamline scientists are most often the ones who work directly with the users. So they have the greatest opportunity to encourage a culture of assessing and documenting beam damage and establishing these good practices among the user community. All right, next I'll go over our thoughts about the future and upcoming challenges. So X-ray sources are tending towards higher flux and more focused beams, which will increase the dose on the sample. And this will require faster hardware and software. Proper beam attenuation strategies will also be needed to incorporate into the experimental stations. It may even be necessary to develop serial data collection strategies. In the most extreme case, it may be necessary to measure each energy point of the spectrum on a fresh sample spot. Alternate sample delivery methods should be considered to minimize the exposure of any one sample spot. And new state-of-the-art beamline should consider implementing simultaneous complementary methods to characterize the sample and monitor beam damage during the course of measurement. There's also a trend towards operando measurements, for instance, measuring biological samples in solution, and this puts constraints on the sample condition. They cannot be measured under conditions that are most amenable to decreasing the beam damage. So in this video, for example, this is a protein measurement in solution, and you can see that under these conditions, the protein aggregates into large clumps. So methods for sample delivery will need to be developed that avoid this type of undesirable behavior. Because of these challenges, researchers need to give explicit consideration to measurement reproducibility, which will require a portion of their beam time. We also strongly encourage collaboration with other X-ray communities like XFEL and crystallography. The scientists in these areas have already thought about some of these challenges, and some of their solutions may be transferable here. So I'll finish with a summary of our recommendations. For users, our recommendations are to know your sample and expected spectral features, prepare the sample in a way that's best suited for measurement, assess beam damage before and during data collection, employ mitigation strategies if necessary, and finally, describe the assessment and mitigation of beam damage when reporting the results. For beamline scientists, our 5 recommendations are to characterize the beam flux density and spot size and shape, develop robust sample environments, incorporate mitigation strategies into the beamline, educate users and encourage good practices, and develop future experimental stations with explicit consideration given to the effects of beam damage. We also had a suggestion to communicate our recommendations using these acronyms: MAP, mitigate, assess, and publish for users, and feed, flux, enable, educate, and develop for beamline scientists. These are conveying the same recommendations, but this is an alternate method of communicating them that people may find more accessible. So with that, I'd like to thank all of the experts who contributed to this work. We had some really excellent discussions over the past couple of months. And I'd like to thank you all for your attention, and I'm happy to take questions.
Okay, thanks, Arun, for excellent presentation. Okay, first we have comments from Simo. Simo, will you speak by yourself? Okay, I will read. Okay. So he said one could also seek out for reliable-based low brilliance tests, at least for ex-situ samples, as one alternative lab measurement. He comments during you were talking in the recommendation for users.
Yeah, I agree. That's a great recommendation.
Okay, go ahead, Professor. Sorry, I couldn't correctly pronounce your name, Professor. Go ahead. Yeah, go ahead. We couldn't hear your. Could you are you speaking? Professor CODO, go ahead. You can speak now. It's your turn. We couldn't hear you. Are you talking now? Okay, we skip him or her and take next questions. Susan, do you speak by yourself?
Sorry, did you want me to read the question?
No, not yet.
Okay.
Yeah, I know the questions, but you can ask by yourself or.
Oh, sure. I was just wondering, just, sometimes people have to do multiple measurements on the same spot, on the same sample, which do you do first? Considerations on that, or also sometimes you have a super pressure sample and it's been through multiple runs of certain measurements. Should we be advocating for a history of that somewhere? Thanks.
Yeah, I think that the measurements, especially if it's a sample that's going to be used multiple times for different types of measurements, this is something that should be considered from the very start. So, for instance, in your question about iron and sulfur, it would be best to start with the measurement that has the lowest chance of beam damage. So that's probably iron in the hard X-ray regime. And I do agree that it would be a good idea to include some details about the sequence of the measurements, especially if there's some possibility that each measurement was done on the same spot and potentially damaged the sample to some extent. But of course, these measurements should be done starting with kind of low flux, using the mitigation measures like attenuating filters to start with conditions that are least likely to cause this beam damage, so that the measurements, all successive measurements, are good quality.
Okay, I will take a question from Professor Teodoro. He would like to know what to do about the samples we do not even know their properties. For example, the environmental, no information about the concentration of elements to measure and so on.
I would suggest then that perhaps the synchrotron X-ray spectroscopy is maybe not the best first step to characterize these samples. It's best if users come, because there's such a limited amount of time, it's best if users come with some information about at least concentration of the metals or what they're expecting. If nothing is known about how the sample will behave in the beam, then it's best to err on the side of minimal exposure, at least to start, and gradually increase the amount of sample exposure if it seems like the sample is not being damaged. So for instance, I would start by using the attenuating filters or detuning the beam to reduce the flux, check the data quality, check for beam damage, and if there is no beam damage, and if the data quality seems to be good over multiple scans, then the user can increase the flux, remove those attenuating filters, and collect their data.
Okay, thank you. It's maybe related to your recommendation for users to assess your sample before coming to beamline, right?
Yes.
Okay, any other questions or comments from the audience? All of you are encouraged to ask.
I
Yeah,
have one
go
quick
ahead.
question. In your experience, is there a minimum flux below which you are kind of safe?
I think this is highly sample dependent, and it's very highly dependent on how the sample was prepared. So I don't think we can consider a minimum flux where there's definitely not going to be any damage. And this is why it's so important to include details like the flux when reporting the data, so that if I'm working on a new sample and I don't know if the sample will experience beam damage, I can at least look in the literature for similar examples and see what conditions those were measured under to try and understand how my sample would behave.
Okay, any other questions, comments, or recommendations? Okay. Michelle, you wanna ask for yourself?
Can you hear me?
Yes,
Yeah,
I
yeah.
can hear you.
Yeah, I just wrote, could you tell us more about the correlation between X-ray energy and beam damage? You were talking that the soft beams is more damaging the hard beam, but if this is now is this correlated with the beam damage mechanism, this would be interesting to know.
Mhm. Yeah, so in the soft X-ray, the absorption cross section is higher, and so there's the sample tends to be more susceptible to beam damage under those conditions because more of those photons are absorbed. I don't know if there's a correlation to the mechanism of beam damage. There can be a correlation depending on if different types of detection methods are used for the soft X-ray. For instance, doing an electron yield measurement versus doing a transmission spectroscopy measurement in the hard X-ray regime.
Thanks.
Diego, do you have a comment? Because you're the reader of the tender X-ray spectroscopy working group. Diego, do you have some comments on X-ray energy dependence and beam damage?
Yeah, hi. Well, we did discuss this as well in our soft tender X-ray spectroscopy group, and well, we are aware that we have seen in beamlines that work with tender X-rays very often some radiation damage that is due to the fact that the interaction is more likely, so that there are more atoms involved, and as well like the fact that you use the electron, but possibly like the total electron yield, you might be extracting electrons that can cause it as well. So we think that from our experience at soft X-ray or tender X-rays, we see this damage happening even more than at hard X-rays.
Okay, thanks. Any other questions, comments? Yeah, Timothy, do you want to ask for yourself?
Yeah, I can just hop in. So yeah, nice talk. It's a very interesting topic to hear. I'm just wondering, you mentioned during the data processing and the interpretation that if you can't achieve a damage-free spectrum, that you can collect damage series and trying retrospectively try and estimate a zero dose spectrum. However, if you have samples that are damaging on a time scale of like seconds or less than seconds, how can there be a certainty that the estimation of the spectrum is what you believe it to be? The spectrum could be completely different. So it's I'm a little skeptical about estimating a zero dose spectrum and publishing that, and you know, it might throw in a little bit of confusion to the community, especially people who are not involved with X-ray spectroscopy often. So yeah, I was just wondering if you could elaborate further, please.
Yeah, definitely. I think that's a good point, and I think some care needs to be taken into how the results are presented if there is some beam damage. So I'll give this one as an example. You can see here that the researchers looked at how certain features changed over the exposure time, and they can estimate based on how, and even the very first scan, how much exposure time that spectrum was taken under, and estimate how quickly the sample is changing at these very short measurement times. And from there, they can at least estimate where this sample may have started out with. You can see in this case, it looks like it definitely started out similar to these vanadium-5 references rather than the vanadium-4 and vanadium-3. And I agree that you probably cannot get an exact representative of what the zero-dose spectrum looks like, but you can still make these arguments based on the changes of what the likely oxidation state or coordination chemistry of the original sample is.
Yeah, I agree to an extent. However, this would then just indicate that you can never be quantitative with this sort of thing. It's always going to be qualitative because, you know, for instance, if you're applying this sort of method to ligand K-edge X-ray absorption and you're trying to quantify the covalency in the pre-edges, then if you're just estimating this, then the values that you get from that peak area is never going to be quite accurate because, as you said, it'll just be an approximation. It's never going to be true because you don't know what the actual true spectrum is. It's entirely, yeah, it's entirely an approximation. So it's if someone is reading this for the first time and using this as a reference to then propose experiments, then it can be it can just be a little conflicting perhaps in the future, but it's just something that needs to, if it's going to occur, and I totally agree that beam damage needs to be certainly cited in papers, and it's something that's definitely a miss right now. But I think a proper log needs to be certainly mentioned in the supplementary information if people are starting to go down this route of trying to estimate zero-dose spectrums. But yeah, thanks for your comments. Appreciate it.
Yeah, absolutely. I agree that it's going to be very difficult to be quantitative, especially if the sample is damaged very quickly. But yeah, that's why we need very explicit discussion of the beam damage in these reports.
Yeah, it can be related to prepare standards for reporting data on beam damage samples. It's a very good idea. Do you have plan to complete these standards? Yeah, this one.
Yeah, so these are our suggestions for what should be included. We wanted to try and establish a minimum inclusion that is accessible and conveys as much information as possible. Of course, we should always strive for including as many details as is needed to reproduce the data. But we think that the suggested inclusion here is a good minimal inclusion that can convey the effects of damage on the sample and how it was assessed during the measurement.
Yeah, it's a very good idea. Okay, any other questions or comments? Oh, there's a question from Daniel. Do you want to ask by yourself?
Yeah, sorry, I was just I just wanted to put a reference in the chat and
Okay,
maybe
sorry. Yeah.
some places that I do actually have a question separately from that.
Sure.
You mentioned a fantastic talk, by the way. Thank you. You mentioned sample rastering as a possible way of mitigating beam damage. And we're in ultrasoft X-ray, so beam damage is really a big issue. And we're planning on implementing some kind of a procedure like this. I'm just wondering if you know if any like beamlines that have implemented that, and are there any special considerations in terms of sample rastering? Thank you very much.
Yeah, I don't know of any spectroscopy beamlines off the top of my head that have implemented sample rastering. But perhaps others on the call are more familiar and can answer this.
Maybe Diego?
I mean, in our beamline, we do change spot, but we don't have a really focused beam, so it's not exactly the same. We have scripts to just collect scans in different spots of the sample. Of course, it depends on the size of the sample, the size of the beam, how many spots you have available. And yeah, I would say it depends also on how quickly you can collect, because if in one scan you already have the damage, then you will not really solve the issue. But I'm sure like there are a lot of beamlines that do raster scanning, and well, I don't know. I mean, there's beamlines that can do mapping as well, so they will be even more.
Yeah, okay, I found a comment from chat. ID26 at the SSRL uses rastering. So there are some beamlines using sample rastering. And next question from Bernd. Do you ask yourself? I mean, it's a comment, yeah. Bernd, are you there? Maybe not. Yeah. It's comment for the previous question. So thank you for your comments. And question from Maria. Do you ask for yourself? Maria Holev, do you ask for yourself, or I can take your question? Okay, could damaging be accumulated with time even when you work with safety beam intensity value at the beginning? Or could it be another mechanisms which affect eventually on collecting data, such as charge effect, etc.? Probably you had a big experience with such type of issue. Thank you. No, Mike, sorry, okay.
Yeah, it's absolutely possible that over the course of many scans on a single sample spot, that damage could be accumulating. So that's why we recommend comparing these many spectra over across a single sample spot or collected on a single sample spot to see if there are changes from the initial spectrum. And also we have a recommendation to move to a new sample spot every so often and collect the low resolution Zane scan, for example. And that way you can confirm that the sample spot that you're measuring and collecting many spectra on is representative of what the rest of the sample looks like, and it's not affected by the exposure time.
Some more questions or comments? Yeah, Edmund? Yeah, do you ask yourself? Oh,
yeah, I will try. I think you hear me. Yeah, I wonder if we should be maybe more strict when we review papers. I mean, this is a problem that's not only affecting beam damage. More often than not today, XAFS is just one piece in the puzzle, and the users don't write much. They write, well, we use the 111 crystal and the beamline in the standard configuration, and that's it. I wonder if we review a paper and we miss this information, should we be more strict, especially if we assume there might be beam damage if it's a sample where typically beam damage occurs?
Yeah, I think that if there's good reason to suspect that there may be beam damage based on similar types of samples, then it's absolutely it should be the reviewer's responsibility to request that the authors address that in the paper. So yeah, I think absolutely that should be something on the mind of the reviewers.
Sorry, there is a continuous discussion on the previous question. Is it only by changes over peak?
Yeah, the changes in the energy or intensity of features is probably the most obvious way that you could see a change in the sample, or sorry, see the effect of beam damage. But some of the mechanisms, like if particles are agglomerating, or if a solution, or if a species is dissolving into solvent and moving out of the beam, that may be visible by, for instance, the edge step, which could increase or decrease. So there's a few features that you may need to keep an eye on. Not just the spectrum itself, but also things like the edge step.
Okay. She said thank you. Okay, any other questions or comments?
Hitoshi?
Yeah.
If you no more comment, question or comment, so I'd like to introduce the email I got from Martin Nakadegal. So he cannot attend this meeting. However, he sent me the email just before this meeting starts, and I put it on the chat.
Okay.
So he said that the one important part of radiation damage, which one does not immediately see, we mean the Martin, we use QuickZAFs to follow the rates of structural change. What we observe is that rates depend on the flux density. So and then he gives the reference. So that is the email I got from him. So it means the QuickZAFs may be helpful to detect the change rate and etc.
It's somewhat related to, yeah, thank you. Go ahead, sorry, go ahead.
Oh no, I was going to say, yeah, thank you for sharing that. I agree with what they're saying here.
Yeah.
Yeah, I think it's related to the slide number 9. The changes occurred gradually, and we can see some changes when we scan repeatedly.
Yes.
Okay, before that, we have another question from Simo. Simo, do you want to ask by yourself?
Yes. You mentioned
go ahead. Yeah, go ahead, sorry.
In the slide 18, you have recommendation for beamline scientists, and point 3, there is mitigation strategies. And I didn't see the lowering temperature, which is, I think, quite common procedure. But you didn't mention, do you think it's still a valid approach?
Yeah, absolutely. Lowering temperature is a valid approach, and that will slow the rate of the diffusion of the radicals. I think I don't have it here, but I have it somewhere in one of the previous slides. But yes, I think that's absolutely a valid mitigation strategy that should be incorporated if possible.
Okay, thank you.
Okay, there's a comment from Simu. For damage assessment at the beamline, staying at the Dane's steepest gradient position and doing a time scan to determine timescales of possible oxidation or reduction may be one fast way to do a first evaluation. Trying to reproduce test scans quickly one after another is slower.
Yeah, I
It's just-
agree with that. And a nice benefit of that strategy is that if the sample is damaged very quickly, doing a quick XAS or Zane scan may not reveal the damage if the damage is occurring even at the first data point. So choosing an energy that is very sensitive, like on the edge, and measuring as a function of time is more likely to reveal if the sample is damaged very quickly.
Yeah. Okay, more questions or comments? I guess no more questions. It's nearly 50 minutes. Thanks, Arun.
All right, thank you.
Thanks for your great presentation. Lastly, Kyoto, do you have some advertisement on the upcoming series?
Yes. Thank you, Hitoshi, and thank you, Arun. That's a session. So I'd like to advertisement for the next. So next Q2XAFS 2024 will be held in October 4th. So they will have a 2 months rest, and they will restart October 4th about tender region X-ray absorption spectroscopy. So today we have some discussion about tender region, and this time the LIGO give us this result of working group discussion. And another one will be held. This one is the 4th, on the October 11th, 1 week later, and that is about scanning type, Q2XAFS, dispersive, and etc. Scanning type and mechanism, and reader is given by the Giantonio. So I would like to have join these two another meetings. And thank you very much for participation. And this